Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. How are you, dear students? Dear students, I welcome you once again in Pakistan International School Thai's virtual learning system, and this is biology class for first year. Dear students, in this lecture we will continue chapter number 4, and in chapter number 4 we will see cellular respiration, some of the topics related to cellular respiration, in which we'll see substrate level phosphorylation, which is done in cellular respiration. Then we will see PGAL and its importance, what is this and how it is formed in the cell, as well as we will see photorespiration. So let's get started. Okay, so if we see phosphorylation, that phosphorylation means adding of, here is as phosphorylation means adding of phosphate in ADP for the formation of ATP, high energy substance or highly energetic molecule, which is required for our daily activities. So in the cell, and especially in cellular respiration, phosphorylation is done by two steps: substrate level phosphorylation and oxidative phosphorylation. So substrate level phosphorylation we will see right now, but oxidative phosphorylation we have seen in detail in chemiosmosis. So substrate level phosphorylation, if we see, that refers to the formation of ATP from ADP and a phosphorylated intermediate rather than from ADP and inorganic phosphate. So what happened in this, that in substrate level phosphorylation, ATP is formed at substrate level from any substrate, from any organic compound, not from inorganic phosphate. Just we can add simply, we can make ATP from ADP plus pi, that is inorganic phosphate. But instead of this, in cellular respiration, substrate level phosphorylation is done. It means directly phosphate or ATP is produced. How it is produced? That inorganic phosphate is not utilized here, but the phosphate is utilized from uh, the intermediate or from phosphorylated substance. How it is done, we will see right now here. So in substrate level phosphorylation, if we see here, that ATP is generated in two steps, that is called glycolysis and Krebs cycle in for substrate level phosphorylation, and that is almost 10% of the ATP which is produced in all this cellular respiration. That is 4 ATPs are produced in this reaction. Oxidative phosphorylation, definitely 90% of ATP is formed almost, that is called 34 ATPs are produced from chemiosmosis, or that is called oxidative phosphorylation. So ultimately we are having 38 ATPs, carbon dioxide and water, as a byproduct of the of respiration process. So here we can say that here enzyme is utilized for adding one phosphate group to ADP, adenosine diphosphate. This one that like that one phosphate comes from the substrate, or that substrate is PE from we can say from glycolysis, and the two steps, and then like in the step number 7, and then we will get sorry, not step number 7, step number 10 in the glycolysis. So then we'll get a pyruvate as the product of glycolysis, and we are having ATP. We can take some examples of them, like how this phosphorylation or substrate level phosphorylation is done. So number 1 is from glycolysis, that is in step number 7, in which 1,3-bisphosphoglycerate changed into 3-phosphoglycerate, means 1 from carbon number 1 from here, 1 phosphate is released out and just added into ADP, then ATP is formed, and here that enzyme 3-phosphoglycerate kinase is used. The other is step number 10, in which phosphoenol pyruvate change into pyruvate by adding 1 phosphate from this substrate phosphoenol pyruvate into ADP in the presence of pyruvate kinase to form ATP. Then the next step is going to be done in citric acid cycle or citrate cycle, so in which succinyl coenzyme A is changed into succinate, and coenzyme A is removed from that, and thiokinase, that is the enzyme which converts this succinyl coenzyme A to succinate CoA, and here GDP with addition of 1 phosphate, inorganic phosphate, we'll get GTP, and then later on this GTP will change into ATP. Okay, so now we will see importance of PGAL. Its name is phosphoglycerate, for that is aldehyde, phosphoglycerate aldehyde, or G3P, that is called glyceraldehyde 3 phosphate. It means at carbon number 3 phosphate is added. So it is having great importance in we can say in Calvin cycle, in photosynthesis, as well as in glycolysis, also in respiration. So in Calvin cycle, that PGAL is called phosphoglycerate aldehyde, that is 3 carbon compound, and that is the starting point for the production of sugar in cytosol in Calvin cycle. So 2 PGL molecule to join to form fructose. We have seen this all steps before, like in Calvin cycle, what is happened, that 2-PGAL can join to form a fructose, and then this is the monosaccharide, and this fructose, this monosaccharide will join to form disaccharide, and then into polysaccharides, and then we can get the starch. In the form of starch, the food is present in the plants. So that is happened when 3-bisphosphoglycerate will change into glyceraldehyde 3 phosphate G3P or PGAL in Calvin cycle, and 1 G3P is released out in the form of fructose, and then we'll get some other, or then it can be converted into glucose, and then we are having other disaccharides and polysaccharides. So PGAL is the end product in the Calvin cycle, that is energy rich sugar, and that is 3 carbon compounds. So when the Calvin cycle once it runs, so it will get 1 molecule of PGAL, and then another cycle run. It means in 2 cycles we will get two PGL molecule, like three carbon compounds, then we can get a fructose or a glucose molecule. So that is very very important intermediate as well, because PGAL change glucose into carbohydrates, or then PGL can be changed into lipids, amino acids, and nucleic acids as well. So in respiration, it is also having a very very important role. Like from glycolysis, it was referred either as PGL or G3P, as we know. So we are not gonna breaking down it in respiration, but in Calvin cycle we are gonna prepare that glucose from G3P. So when glucose is broken down and changed into fructose, a 6 phosphate, then in glycolysis it is changed into two compounds. One is PGAL, that is called G3P, and other is dihydroxyacetone phosphate. Now this is unstable compound, and it can again change into PGAL, and then will proceed the next reaction, that is called payoff phase, in which we'll get ATP and NADH as well energy, and we get a pyruvate. So this is performs PGL performs an intermediate uh, product or intermediate, we can say, the compound present in respiration. Cellular respiration of fats and proteins, that we know that not only carbohydrates but fats and proteins can also be used as a fuels in respiration. So how this process is done? So we can see here that carbohydrates, when they are digested, they form into the glucose, and then this glucose is what, a 6 carbon compound, and then it will break down into 3 carbon compound, that is called G3P, on that the same PGAL. Now this PGAL converted into pyruvic acid, and then it is transformed into acetyl coenzyme A, and then it enters into the Krebs cycle. This is the reaction of or breaking down of carbohydrates. But fats can also be used here in this reaction for giving us energy, and that can fats can be utilized to break down into glycerol and fatty acids. You can see here that it is utilized for uh, like the units of fats are glycerol and fatty acids, so they are broken down into this one. So this glycerol will change into glyceraldehyde 3 phosphate, like PGAL, and then again it will move into the Krebs cycle. And here fatty acids also just broken down into acetyl coenzyme, that is 2 carbon compound, and then it will again directly move into the Krebs cycle. Here the protein can also be broken down into amino acids. We know from here amino group is released out, and it will change into keto acids. These keto acids can be directly move into the Krebs cycles, or it can just change into pyruvic acid, and then pyruvic acid just move into the Krebs cycle. This amino group will move to the liver, as we know, for deamination, as we know that liver performs the function of deamination. Okay, up to here we have discussed all about respiration, but there we are gonna see another term of respiration, that is photorespiration. What is photorespiration? That it is also known as oxidative photosynthetic carbon cycle, or that is also said to be C2 cycle or C2 photosynthesis. In plants specifically, uh, which attempts to reduce the consequences of wasteful oxygenation reaction by enzyme Robisco. So here this is very important enzyme which is going to like going to perform its function here, or the process that leads to only one 3-PGA being produced in the dark reaction in chloroplast. Actually, this is related to the photosynthesis process, but here photosynthesis does not occur properly, and that is called photorespiration, because in like respiration we consume oxygen. So here also plants consume oxygen in place of carbon dioxide. That's why it is called photorespiration, because it is done in the daytime. So like it is now known that this response to oxygen is due to the competition between oxygen and carbon dioxide on the Robisco enzyme. So this is very important enzyme which performs this function, and that is also called Kelvin Benson cycle, because these two scientists have discovered this process. In normal photosynthesis, what happened? Carbon dioxide is joined with RUBP, ribulose bisphosphate, to form two molecules of 3-PGA. Then this PGA, like yes, joined with carbon dioxide, and then we'll get our required product. But here you can see here this one. So this Robisco is going on properly, and two molecules of 3-PGA are formed. Robisco means ribulose bisphosphate carboxylase and oxygenase. So it means that it performs the function for both of the processes. Like one is carboxylation, like addition of carbon dioxide, the other is oxygenation. So photorespiration occurs when Rubisco carbo or RUBP oxidase joins oxygen to RUBP in the first step of Kelvin cycle rather than carbon dioxide. So like this is just, it seems to be very minor change, but that having the great difference in the product. Whichever compound, oxygen or carbon dioxide, is present in higher concentration, so Rubisco joins that one with the RUBP. So photorespiration prevents the synthesis of glucose and utilizes plants ATP, because in the presence of oxygen, what happened, energy is formed, and then it is being utilized. So here glucose is not being produced properly. It prevents a synthesis of glucose, because it is not joining carbon dioxide, it's joining oxygen here. So if more carbon dioxide is done, so Rubisco joins with carbon dioxide and photosynthesis occurs. If more oxygen is there, so Robisco joins with oxygen, and more there will be less photosynthesis. So here you can see that here this is the normal reaction in which carbon dioxide is joined with RUBP in the presence of Rubisco, and the normal function of Kelvin cycle is going on. But here oxygen is adding here with RUBP in the form of Rubisco. So there is very toxic substance is produced, that is called 2-phosphoglycolate. 2-phosphoglycolate is very dangerous for the plants. So plants have adapted according to that, and they change in 2-phosphoglycolate into glycine, then serine, then glycerate, and then again RUBP is formed. Because this glycolate is very dangerous or harmful chemical for the plant, so quickly, within no time, it has to be changed into some other less toxic substance. So in photorespiration, what is happened actually? It is done in if the problem is water stress, and the plant is under water stress. So what happened? They close their stomata in the daytime. So for what reason? To prevent water loss through respiration. Here, when they close stomata, so the limits gas exchange, so it means oxygen is still being produced by the process of a photosynthesis through light reaction. The concentration of oxygen is increasing in the cells, but carbon dioxide is not entering from outside of the leaf, from the stomata, because stomata are closed. So this is actually the main reason, as the carbon dioxide is being used up, which is used in the Calvin cycle. So oxygen is continuously increasing and carbon dioxide is continuously decreasing. So this is the reason when carbon dioxide is increased, so this rubisco use this oxygen to bind with. So this whole process is done in, we can say, the three steps or three organelles of that of the plant cell, to save them from like from a more destruction in the plants. So what happened from here? If you see from upper side, oxygen is adding in the chloroplast. As we know that oxygen is added in carbon dioxide, or photosynthesis is done in chloroplast. So when oxygen is increased in the chloroplast, so Robisco adds oxygen with the chloroplast, in the chloroplast, and then it is changed into phosphoglycolate plus PGA. Now this phosphoglycolate, because that is very toxic, so it will soon change into some other compounds. So it will change into release one phosphate from here, and it will change into not phospho, but just glycolate. Now this glycolate will enter into peroxisomes. Now peroxisomes is having the great role in the eukaryotic cells for, we can say, the oxidizing organelle. We can say this is the oxidizing organelle, in which what happened, that hydrogen is oxidized and changed into hydrogen peroxide. That's why its name is peroxisomes. And this is hydrogen peroxide is added in glycooxylate, and then it will change into glycine. A hydrogen peroxide is formed by adding here into glycolate what more oxygen, and this oxygen just takes a hydrogen or proton from glycolate and change into hydrogen peroxide and removed out, and then it is broken down into water and oxygen. And this change into glycolate change into glycooxylate. This glycooxylate then change into glycine and form this enters from here, that glutamate, and then it will change into alpha ketoglutarate. Then glycine will enter into mitochondria. In mitochondria, this glycine removes ammonia from it, and as well as carbon dioxide, to just change into serine. And now from mitochondria, this serine will shift into peroxisome. In that reaction, a reduction of NAD is done, and then we'll get NADH. Then this serine, as we know that ammonia will go for further detoxification, and carbon dioxide will be definitely increase in the cell. When carbon dioxide will increase, so then Robisco will just again join RUBP with this carbon dioxide. The serine will enter into peroxisome, and then it will change into hydroxypyruvate. Hydroxypyruvate, because here mm, and then enter glyoxylate will enter, and it will change into glycine. So this will form hydroxypyruvate. This hydroxypyruvate change into glycerate, and it means from here hydroxy, the hydrogen is added into NADH is oxidized again into NAD plus, and then we will get glycerate. Then this glycerate will again move to the chloroplast, and then here ATP is utilized, and glycerate will change into PGA. Actually, this PGA is that compound which just move on into the Calvin cycle for to make RUBP. So we say that this is energy consuming cycle, because here ATP is utilized, and as well as here NADH power is also utilized. So this is the whole of the cycle or reaction is done by the plant cells to just save them from glyoxylate and glycolate, which are the highly toxic compounds which are present which are formed by the combination with RUBP of oxygen and in place of carbon dioxide. So dear students, that's it for today. Inshallah, we will see you in the next lecture. Wish you all the best. Allah Hafiz.